Good evening, all of you. And today's lecture we will be discussing about the production technology of banana. Banana is one of the important tropical fruit, and in the net ICR net SMS STU and ARS exam. In the tropical fruit production, there are these are the important tropical fruit crops: banana, mango, papaya. And from these crops, there will be questions for sure. So we will be seeing the production technology of banana in detail. So here the family of banana is Musaceae and the order is Zingiberales. And the chromosome number, it can be 2x, 3x or 4x. And accordingly it will be 22, 33 and 44, and the edible portion is mesocarp. The fruit type is berry, and this is the banana is the first fruit that human have started cultivating. And in the food commodities, if you look at the world, banana and plantain are the fourth important food for the world, and it is also staple food in the African countries like Uganda and Tanzania. And this is also one of the most traded tropical fruit in the world. And in the world trade, the main banana varieties are dwarf cavendish, granine, that is spelling mistake for granine, you can check, NAINE, robusta and lefton banana of Philippines. And the largest diversity of banana is observed in southern Asia, and we will see how it reached all over the world. Arabs introduced banana from India to Palestine and Egypt in the 7th century, and from the east coast of Africa it reached different countries of the Africa. Then from Canary Islands to it was introduced to Central America in 1516 AD, and to West Indian Islands, Christian missionaries introduced the banana. Then coming to the origin, so there are two species, Musa acuminata and Musa balbisiana. So Musa acuminata, the primary center of origin is Malaysia, and for Musa balbisiana, it is native to India and Burma. So this Musa acuminata was introduced to India and Burma, where Musa balbisiana was a native species. And there in the Indo-Burma region, the natural hybridization of the two species occurred, and we have got the edible bananas. And because the natural hybridization occurred in this area, India has got the second largest diversity of bananas in the world. And there are more than 600 types of germplasm together, and among that India has got more than 300 out of the 600. So that's why we see India is the second largest diversity of bananas in the world. So all the banana and plantain which are cultivated now are land bases, uh, which are farmer selection from natural intra and interspecific hybridization of the two different species which we have discussed. Musa acuminata, which is the donor of AA, and Musa balbisiana, which is the donor of the BG. And edible banana is native to warm, moist regions of tropical Asia, Siemens 1962. So there are two genera, that is Musa and Ensete. Ensete is a all declining genus, and it is originated in Asia, and it is having about 6 to 7 species. And among these different species, Ensete ventricosa. Ensete ventricosa is reported to be grown in Ethiopia as a food crop. So Ensete is an old declining genus, and it is originated in Asia, and it is having 6 to 7 species, and out of that Ensete ventricosa is reported to be grown in Ethiopia as a food crop. The next genus is Musa. And Musa genus is about 50 species and they are divided into 5 sections. So the 5 sections are Eumusa, Rhodoclamis, Calimusa, Australian Musa and Insectivorous. So Eumusa, it includes 13 to 15 species of edible as well as wild banana, and the chromosome X11 and 22 and 33 is also found. Then 44 is found very rarely. So the example of species coming under the section Eumusa, Musa acuminata, Musa balbisiana and Musa basju. The next section is Rhodoclamis, X equal to 11, mostly diploid. It is seen from India to Indonesia. 5 to 7 species are kept. And here Parthenocarpy is absent. Example, Musa ornata and Musa velutina. Musa ornata and Musa velutina. This comes under the class Rhodoclamis, under the section Rhodoclamis. In Calimusa, it is a fundamental value x equal to 10, 20, present in Indochina, Malay and Borneo regions. Same like Rhodoclamis section, here also Parthenocarpy is absent and it includes 5 to 6 species. Example, Musa coccinea. The next section is Australian Musa. Australian Musa, chromosome number x equal to 10 and 20. And this is found in Queensland, that is in Australia, and also in Philippines. And here in this section, fruits will be developing parthenocarpically. Then Musa textilis or Manila hai, Musa maclavi and Musa freehi. These are the important species coming under the section Australian Musa. Then Insectivorous, it includes Musa ingens x equal to 7, 2 and 14 of New Guinea, which grows to a height of over 10 meters. So this Musa ingens is the largest non-herb. This is the largest non-herb, and another species in this group is Musa beccari X9 to 18, and this is from North Borneo. The most important Musa cultivars are almost sterile triploids 2 and 33, and the Simmons and Shepherds in 1955 reported a scoring technique to indicate the relative contribution of the two wild species for the constitution of a given cultivar. So they have made 15 distinguishing characters between Acuminata and Balbicinia, and if a character is close with Musa acuminata, the score of 1 will be given, and if a character is close to Balbiciana, score of 5 will be given, and intermediate expressions will be given scores of 2, 3 or 4 depending on their intensity. So if there is a score of 15 to 21, it can be, the genomic constitution can be AA or AAA. Then if it is 26 to 41, AAB. Then 48 AB, 59 to 65 AB. So this is the taxonomic scoring. So here the characters are given in the first column. Then first Musa acuminata and then Musa balbiciana. So these are the characters: pseudostem color, petiolar canal, peduncle, pedicel, obvious bract shoulder ratio, bract curling, bract shape, bract apex, bract color, bract scars, free tepal of male flower color, and stigma color. So as we have discussed earlier, this system, genomic scoring system, was given by Simmons and Shepherds in 1955, and if a character is close to Musa acuminator, we will be giving a score of 1. And if it is close to the character of Musa balbicinae, we will be giving a score of 5. And depending upon the intensity, we will be giving 2, 3, and 4 for in between intermediate characters. So this is the taxonomy classification of edible banana proposed by Siemens and Schapolz in 1955. So here you can see first column there will be genomic constitution, and then the ploidy level and the score and nomenclature or the which are the banana groups coming in each group. So AA, that is ploidy level is 2x. So the score will be 16 to 23. Examples Matti and Anakumben. Then AAA, that is ploidy 3x. 15 to 21 will be the score, and the bananas coming in this group Gros Michel and Cavendish. Then AAAA, that is ploidy level 4x, and the score is 15 to 20, Bortless Altafort. So this is a synthetic hybrid developed in West Indies. Then AB 46 to 49, varieties coming under this group Neipuvan and Kundan. Then AAB ploidy level 3x, genomic scoring 26 to 46, Champa and Rastali. The Rastali there is spelling mistake. Then ABB, the ploidy level is 3x, 59 to 63 will be the score. Kanjikela, there is also spelling mistake, I is missing. Then Mountain. Then ABBB, ploidy level 4x, and the score is 63 to 69, and the variety coming under this is Clue Tepero. So that's all about banana. We have just discussed about the introductory part of banana, like the origin and also the genomic scoring. So we will discuss the remaining production technology in the next class. Thank you.